Hello everyone, welcome to another lecture of drug delivery engineering and principles. Just a quick recap of what we learned in the last class. First we talked about intra-articular injections. So, these are injections which are given into the joint space. So, if this is my femur, this is my tibia, you can directly inject things in air and that will result in much higher residence time. What we also learned is this synovial membrane contains lots of blood vessel and lymphatics, and they can clear out the things very rapidly if they are smaller size. So, if you want to inject something to reside very for long durations, you want to inject bigger particles that can release drugs, or the drugs itself may be big. So, somewhere anywhere between above 1 micron would start to give a much higher residence time than anything below that. Then we started IV administration, one of the most widely used methods in clinics, especially for things that you want to have instant action on the body. We then discussed various organs that put the limit on a particle size. If you want to do a control release through some bioengineering approaches, we discussed that first of all kidney will clear things below 6 nanometer, so or below 10 nanometer. So, you want the particle sizes to be above 10 nanometer. Then we said that anything with the size above greater than 200 nanometer, the spleen will clear it away, 200 nanometer plus particles. So, you want the sizes to be below 200. Of course, the biggest blood vessel is 5 micron, the smallest blood vessel, sorry. So, you should definitely have less than 5 micron, and then we also discussed that liver is another major organ that is going to clear quite a lot of your big particles. So, basically you want something in the size range of 10 to 200 nanometer. Okay. Then we talked about EPR effect in both tumor and inflamed tissue, and what it is the blood vessels that are feeding your tumor tissue are fairly dilated, whereas the blood vessels in your healthy tissue are fairly open. So, this is healthy tissue, this is tumor tissue, and these pores are about 100 nanometer in tumors, and they become larger as the tumor grows, as well as there are also pores in inflamed tissue when the vessel dilates to allow more cells and more nutrients to go in. And because of that, if you have size ranges in between 10 to 100 nanometer, you will have a much higher efficiency of delivery, and this is called the enhanced permeation, because they will go here but not here. And then this tumor microenvironment also has very poor lymphatic system, so it can't really clear things away once they go in, and that is enhanced retention. So, which is nothing but EPR effect. So, that is something you can utilize as well, utilize quite a bit in the literature. Okay. So, let us continue our discussion on the IV route that we were talking about, and we are going to talk about a drug called Doxil, and this is similar to the EPR effect. It utilizes that effect and is an approved nanocarrier for cancer drug delivery, and Doxil contains doxorubicin as the major effective drug. The doxorubicin is a very effective anti-cancer drug. In fact, most initial cancer studies are done with doxorubicin, and but the problem is it has a very high cardiotoxicity. So, cardiotoxicity means that something related to the heart. So, it starts to kill heart cells, and then obviously when you inject something IV, it is definitely going to go to the heart as well. So, that is a problem, and that limits the amount of dose of doxorubicin you can give to your patient, because eventually at a certain dose all the tumor cells will die, but before that dose is reached the patient may die because of all this cardiotoxicity and even other types of toxicity. So, that puts a limitation on the dose of doxorubicin that can be administered. So, to counter that, what was done is the doxorubicin was taken, it was encapsulated into a liposome, This was done through reverse loading. I hope you guys remember what reverse loading is. So, reverse loading is nothing but using the property of molecules to diffuse into the lipid membrane if they are non-ionic at a certain pH, and once they go into the membrane you make the liposome such that the inner environment has a different pH and they will become ionic. So, that will cause the movement of the drug to go into the liposomes and effectively build up a very high concentration in the liposome. So, that is what was done, and then these were actually further pegylated. So, again I hope you remember pegylation. Pegylation prevents the interaction of various cells and protein with your biomaterial surface, and because of that it will not let this particular liposome get absorbed with several proteins or get cleared away by the immune system. So, it increases the residence time, and this resulted in quite a bit effectiveness of this. It was used very heavily for ovarian cancer, still used, and it also reduced the cardiotoxicity, and because this was using an EPR effect. So, what happened is this was made of size of about 100 nanometer, and because of that the vessels in the heart were fairly healthy in tumor tissues. So, this liposome was not going in and releasing doxorubicin in the to the heart cells, but then in the tumor areas where the vessels were fairly dilated and these particles can go and permeate, these were accumulating in the tumor areas, and actually effectively what you did is you, let us say if you had a ratio of drug in tumor versus the drug in healthy tissue, that for your free drug was much less than for your doxil, and because of that this therapy took off and was used in the clinics as well, and is still used in the clinics. So, here is also showing that not only that, you actually improved the pharmacokinetics also, because doxorubicin is a very small molecule, much less than 6 nanometer. So, doxorubicin gets cleared away from the system very quickly as well. So, you have to continuously infuse doxorubicin in the patient if you want any therapy, whereas doxil, since it is now about 100 nanometer, its residence time is very high and it slowly releases the doxorubicin into the system. So, as you can see, the concentration of dox was almost in fact more than 2 orders of magnitude higher than what you had in cases of free drug. So, here is another example. So, this example, the product name is Abraxane, and this is basically one of the vials that you can find in the market, and what it is carrying a drug called paclitaxel, again very effective against lots of cancer cells, especially breast cancer, but it actually has a very low solubility in water. So, what that means is, and it is fairly small molecule. So, what that means is you can't really inject too much because it will precipitate out and again may clog your heart vessels or your blood or your brain vessels, causing heart attack or stroke respectively. So, what was done is this paclitaxel molecule was conjugated to albumin protein, which is one of the most abundant protein in our blood, and it then self assembled into a structure, because paclitaxel is fairly hydrophobic and it did not really wanted to interact. So, it self assembled into a structure like this, where paclitaxel ended up being in the core and albumin changed its conformation to reflect on the outside and interact with water with a mean size of about 130 nanometer, again close to that EPR effect range that we wanted, and that resulted in very enhanced and increased transport and accumulation in tumors. So, this drug was approved in 2005, had bumper sale that year, and still is being used quite a lot. So, as I said, it is nothing but a protein bound drug that you are using and forming a nanoparticle out of that and using the EPR effect that we talked about. Here is another example. This, in this case we are looking not at tumor, but at inflamed sites or infectious site. So, the product is called bisosome, and what it is again a liposomal formulation in which the drug being used is amphotericin, which is an antifungal drug, and but then this this drug causes lots of side effects in nephron, basically kidney. So, that was causing limitation of how much drug you can use, but what these folks did is they encapsulated this or actually attached it to the surface of the liposome into the lipid membrane layer. That caused first of all increase in the size, so it did not really accumulate it in the kidney, and the secondly controlled release as the liposomes release this drug out. So, this was the product, and that was approved in 1999 and again was used quite often. Okay, so this concludes our session on the route specific delivery. We are now going to change and start a different module, which is immune response to materials, and so let us get started on this. So, if you have any material that you are putting in, and we have talked about this briefly as we went down through this course, but if you are introducing something new, the body is going to identify this as foreign material and there will be some immune response against it. So, what we will do is we will try to understand what kind of immune response builds up in these circumstances, and how do we tackle this, how do we make sure that this immune response is not rejecting our material that we are putting in, it is not affecting the performance of whatever the function the material was supposed to do. So, this is what we are going to learn. So, before we are able to learn that, we have to learn some a little bit immunology that will be required to understand this module. So, let us start with this. So, this is a very classic figure, and this is taken from Janeway, and this is showing different cells of the immune system. So, you have a bone marrow which is located in the long bones, it is the cavity within the long bones that you can find, and that is a site which is extremely rich in pluripotent hematopoietic stem cells. And what does this pluripotent mean? That means that these stem cells can give rise to several different cell types, and so these give rise to several different cell types, as you mentioned. Some are called common lymphoid progenitor, some are called common myeloid progenitor. The myeloid progenitor goes in and further differentiates into granulocytes and macrophage progenitor, including some of the blood cells as well, and then they further go in and differentiate into blood cells and platelets, whereas the common lymphoid progenitor has a much more complex route. They go and differentiate into B cells and T cells. Again, there will be a lot of biology, but bear with me and we will describe these as we go along. So, you have B cells and T cells, and then they also give rise to some of these granulocyte, which was from the myeloid progenitor, will also give rise to neutrophils, eosinophil, basophil, and there are some unknown precursors as well, monocytes, dendritic cells. These are all different types of immune cells which are responsible for different functions, and we will describe some of them as we go along through this module. The B cells and T cells can then have different types as well. They can have effector cells or they can have helper cells. So, both B and T type of cells can have both effector and helper cells. So, some of these B cells are responsible for secreting antibodies, something that I am sure you must have all have heard by now, and then there are T cells which are directly responsible for killing any rogue cells or any cell that is not functioning well and immune system is not liking it, and some of these monocytes and granulocytes derived things can then go in and become mast cell, macrophages and dendritic cells, and these are cells that are more responsible for presentation of anything to the immune system which they think is foreign. And then there are lymph nodes, which are nothing but they are these. So, I said that there are blood vessels, Then along the blood vessel there is another parallel network that is called lymphatics. So, let me just take another color for that. So, along with the blood vessels there is another parallel circulatory system that is called lymphatics at a much lower flow rate, but nonetheless throughout our body, and this lymphatics from time to time and from different places is actually harbors a secondary lymphoid tissue which is called lymph nodes. And what these tissues are is a depot of these immune cells which are just sampling around. So, and obviously this is tissue. So, what it is they are sampling whatever is coming out from this tissue going into the lymph node where immune cells are present in very high numbers. All these B cells, T cells and other types of macrophages and dendritic cells are present in very high number, and they are continuously signaling as to if there is something foreign. So, let us say if there is a bacterial protein that is going to come in here, it is going to be picked up by one of these immune cells, particularly dendritic cells and macrophage, and it is going to be then presented to one of these effector cells or memory cells or the helper cells, which will then mount an effective immune response against it. And again if you are unaware of this, do not be worried, we will describe it in a little more detail as we go along. So, these are some of these common terms, cells of the immune system. Most of them are actually fairly important for this module. So, it will be good if you can start to remember some of these cells. And so, let us talk about function of these cells. So, macrophages, which again we have discussed before already, these are cells that take up foreign objects, foreign bacteria, foreign viruses, and their major job is to first of all do phagocytosis, which is a process through which large particles and large foreign cells are being taken up, and then they get activated and start secreting lot of bactericidal enzymes and bactericidal proteins that kill them. And then they are also responsible for antigen presentation, which means that they are the ones who, even when they kill them or even when they harbor them, they will present it to the rest of the immune system saying that, hey, look, there is something foreign here. So, that is the major role for these. Dendritic cells like macrophages are again very similar, and they are also responsible for phagocytosis and antigen presentation. Neutrophils are slightly different in that regards. They do a lot of phagocytosis too, however they are not as involved in antigen presentation as the macrophages and dendritic cells, but they are mainly responsible for killing the bacteria. So, they even act as a kind of a suicide bomber where they carry lots and lots of harmful bactericidal molecules, and when they find that there is bacteria in the surrounding, they will just burst and release all that near the bacteria vicinity, killing the bacteria in the process. Then we have eosinophils, and one of the function responsible for them is killing of the antibody coated parasites. So, they are more responsible for big worms like pathogens, and they can even be effective against some of the smaller pathogens too. And these basophil mast cells not as widely studied and understood. Probably their function in the immune system is either not very understood very well, or they do not really play a very major role, but their major function is to cause inflammatory reactions, and they are actually also responsible for allergies. The same with the mast cells, they also release granules containing histamine and other active agents to kill the surrounding environment, bacteria and pathogens. Okay, so how does the inflammation start? So, this is important because we need to understand how does it start, so that when we put a material we make sure that this does not happen. So, let us learn first of all how does it start. So, again the immune system is very vast and contains lots and lots of cells, and they are surveying pretty much every part of the body that we have. So, all tissues that we have contain some one or other type of the immune cell. So, what happens if there is some injury, these cells become very active. So, they will then go to that injury site, they will sample that to see if there is anything that is foreign that caused this injury, why did the injury happen, and if they do find, they will continue to digest this foreign antigen, and the cells that are dying there, because obviously if there is some injury, then you must have some dead cells, your skin may have ruptured, so skin cells are dead, some of the blood vessels may have ruptured, so those endothelial cells may be dead, and if they do find something that is causing this, they will secrete cytokines. Sometimes, I mean, most of the time they will even do that without anything, the initial injury is enough for them to start secreting some cytokine. So, let us say if this was macrophage that was sampling that area, which is one of the major cells responsible for also sampling tissues. So, then they will start secreting several classes of these cytokines, which are and we will talk about it in the next slide, but which are nothing but protein based molecules involved in signaling and amplifying the immune system. So, some of them being IL-1, TNF alpha, IL-6, IL-8, IL-12, these are some inflammatory cytokines, and they all have their own functions, which some of them are overlapping, some of them are systemic. So, they will actually involve the whole immune system of the body, some of them are local, which is more at a localized level. But what will basically happen is like an IL-1, if it is released, it is going to cause activation of the vascular endothelial, it is going to activate any kind of lymphocyte, which is a type of immune cell in the surrounding, cause the local destruction of the tissue, and will even cause the increase in the effector cells, which are the cells which are going to kill anything foreign or going to help in eradication of that to come into the site. So, another class of molecule is TNF alpha, similar to IL-1, it is fairly inflammatory, it activates again the vascular endothelial, increases the permeability. So, if you remember in the tumors part of the talk, I was talking about that if you have inflamed tissue, your again vessels are more permeable, they are not as tight as they are in healthy homeostatic tissue, and that is because these factors are being released which are signaling these cells to increase their junction diameter, and what that does is it causes more cells to come in, more growth factors, more immune cells, more cytokines to come in, and that is how the inflammation is more leakier, the inflamed sites are more leakier, and so that causes entry of antibodies, complement different cells into the site. Then we have IL-6, which are again responsible, all of these IL-6, IL-8, IL-12, these are responsible for again activating different immune cells, some of them NK cells, some of them causing antibody production, TH, which is the T helper cells, they are all essentially accumulating at the site. So, I mentioned cytokines and chemokines, let us quickly define what these are. So, cytokines are small proteins of typically about anything between 25 to 30 kDa, released by various immune cells. So, typically this is immune, although other cells can also release them, and they are usually released in response to a certain stimulus that might be given, and then they induce responses by binding to a certain receptor. So, lots of cells have receptors for these cytokines, and they go and bind to that receptor, which then signals the cell to either get activated, to start migration, to start causing the vessel dilation. So, all of this is controlled by these cytokines, and there are several classes of them. So, one thing as we are going along I like to point out is immune system is fairly a dynamic research topic. Every 3-4 years the whole immune system understanding changes by quite a bit amount. So, what I am giving you now is what is currently well accepted and well hypothesized and in the books, but all of these are fairly lucid definitions and they may change as the time goes on. Okay. So, these cytokines can act in an autocrine manner, which basically means that they can act on the cells that are actually releasing them. So, let us say if this is an immune cell and it releases this cytokine out into the media, the cells themselves have receptors for these cytokines, and when these cytokines bind to the same cell, they will activate the cell further. So, this is kind of a positive feedback loop where cell is getting activated, it is releasing these cytokines are binding to the cell itself and activating them further. or they can act in a paracrine manner, where once let us say these cells have released it, there is another cell in the surrounding that goes and binds to it, and this cell now gets activated. So, now the signal is amplifying because the neighboring cell is also becoming inflamed. So, and then some of these cytokines can act in an endocrine manner, where that means that let us say a cell has released it in a certain organ, that cytokine is now going into the blood vessel, and that blood vessel is taking it everywhere in the body, where to even distant parts of the body it is causing this thing to activate the cells. However, obviously since now this is going to involve some time as well as exposure to lots of different proteins, this will depend on the ability of the cytokine to first of all enter the circulation, and then finally once they enter the circulation, then how stable they are, what is their half life in the circulation, because if the half life is very low, then they will not be able to go ahead and do activation at a distant organ. And then there are chemokines. Chemokines like cytokines are also secreted by immune cells. They are actually a type of cytokines. So, chemokines are part of a cytokine, and then they act as a chemoattractant property. So, what that means is, let us say if there is a problem at this site or this cell and it is secreting some of these cytokines, which are then building up in the surrounding. So, obviously what will happen is, near the cell you will have quite a bit, away from the cell it is going to become less and less. So, let us say there is another cell bit far away from this cell, but as it secretes that more and more cytokines are coming from this direction, this cell will then move and get closer to this. So, it is acting as a signal to call more immune cells to come to the site. So, that is what chemokines do. So, let us talk about some physiological events. So, let us say if there is a tissue injury and some foreign entity is also present. So, what is the first thing that is going to happen, that the tissue macrophages and the neutrophil will get activated. So, neutrophils are not really resident a whole lot, but they are in quite high numbers in the blood, and if there is a tissue injury, then you have also ruptured a blood vessel. So, neutrophils are present at the site. So, they start doing phagocytosis to whatever was damaged and out in the surrounding. If they get activated, they will start to secrete cytokines. Now that they are secreting cytokine, this local cytokine release is going to trigger a cascade of events, right, because as we discussed in the previous example, first of all these cytokines are going to act in a feedback loop mechanism, either on the same cell or in the neighboring cells, and amplify their production. They will start calling immune cells from the surrounding. So, more immune cells are now coming in. So, more immune response is being developed, and as well as some of them will actually go far away and start activating the immune cells there as well. And some of the other cascade of event is, first of all, vasodilation. So, now they will also dilate your blood vessel, which means that more immune cells can come into the site, and that is why you see actually that site becomes fairly red. That is because the blood circulation has increased there. Then you have influx of protein rich fluid and the cells are coming in. So, now you see swelling as well. Now, you have lot more fluid into the environment where the injury has happened. So, that is why you see swelling, and that causes pain as well, because nerve cells also get involved. And then you have influx of leukocytes and phagocytic cells into the tissue. So, because of this vasodilation, all these phagocytic cells are also, again, which were circulating in the blood, are also coming into the tissue. And then if the next step is, if you are able to clear whatever was a foreign entity and the body thinks that this is fairly compatible, then the next step is healing and tissue regeneration, which in cases of biomedical is what we want. So, more new vessels will form, and maybe the scar tissue will form if it is a deep wound and all, and the healing process will start. So, what are the systemic effects of the cytokines? So, the released cytokines can act on distal tissues to further facilitate immune response. So, this is on the systemic effects of it. So, some of these proteins like IL-1, IL-6 and TNF alpha can potentially do that. So, if they go to liver, they will activate some other proteins like C reactive protein and menin lectin. This will leads to an activation of a complement system. We have not talked about complement system yet, I think we will talk about it in the next few classes. So, just hold on to this, but it activates another class of an immune response that we have not talked about yet. The other thing that they will do is in the bone marrow, they can activate the bone marrow endothelium and they will start mobilizing more neutrophils. So, as I said, bone marrow is the site where all these immune cells are being produced. So, they will start production of neutrophil and start to cause them to come to the site. Then they can also go and act on hypothalamus, which is a brain organ, and that is responsible for increasing the body temperature. And the reason it is done is because our immune cells are actually, and our own cells are actually much more capable of handling a higher temperature than let us say a pathogenic cell. So, if you increase the body temperature, so from 37, let us say if you go to 39, the body is at a more advantage at that point than these pathogenic cells. Then similarly, the fat and muscle tissue also get these signals and they start, because now you will need some energy to maintain that much heat, to have that much influx of cell, to have more cells come in. So, that will start happening, and again both of this will cause decrease in viral and bacterial replication. And then more dendritic cells are being called in, again these are also cells that are going to do phagocytosis and make sure that your bacteria is being killed. Okay, we will stop here and we will continue in the next class.